I'm Missy Jenkins, and I serve as Vice President of Public Policy at the Alliance for Aging Research, and I am the Executive Director of the Accelerate a Cure and Treatments for Alzheimer's Disease Coalition, or ACT-AD. On behalf of ACT-AD, it is my pleasure to welcome all of you and our presenters, Dr. Rudolph Tanzi and Dr. Rob Moir, to our first webinar of 2019 entitled The Emerging Antimicrobial Protection Hypothesis of Alzheimer's Disease, which was published in October. By way of background, ACT-AD is a group of more than 50 national nonprofit organizations working with the FDA, other government agencies, industry partners, and policymakers to increase attention and resources devoted to the challenges posed by Alzheimer's disease. Until ACT-AD's formation in 2005, there was no single point of advocacy that combined the perspective of advocates for research funding, aging services, and caregivers. The FDA has taken several actions in response to ACT-AD's advocacy, including one, establishing a neurology interagency working group across the FDA to pool expertise and collaborate more efficiently on the evaluation of promising new treatments. And two, the FDA has also extended its patient representative program to Alzheimer's patients and caregivers. And finally, three, the FDA began working with the coalition on an annual workshop, our annual ACT-AD FDA Allies meeting, where representatives from the FDA could interact with leaders in the patient advocacy community, academia, and industry on issues that create roadblocks or potential roadblocks to the successful review of new Alzheimer's disease products. This past December was our 11th annual FDA Allies meeting, where we focused on the promise and challenges of big data approaches for biomarker discovery, drug repositioning, and combination therapy development for Alzheimer's disease. That white paper that we produced as a result of that meeting will be published on the ACT-AD website later this week. Now for a few housekeeping items. Our presenters are going to speak for about 40 minutes, followed by Q&A. As you think of questions, please use the WebEx chat function. We're recording today's program, and it will be available within a few days at the ACT-AD website, which is act-ad.org. I'd like to thank all of my colleagues for their help in planning the program. Sue Peshin, the Alliance President and CEO, Sarah DeGiovanni, Brandon Miller, Ryan Carney, Lauren Smith-Dyer, and Janelle Germanos. I'd also like to thank Phil Cronin, who helped us put together today's webinar. Finally, I'd like to recognize ACT-AD sponsors: Alzheimer's GermQuest, Anavex, Avenir, Biogen, Eisai, Eli Lilly, Genentech, Janssen, Lundbeck, Merck, and Otsuka. Without your support, programs like this one would not be possible. Thank you for your continued investment in developing a cure and treatments for this devastating disease. Now, I have the pleasure of introducing our speakers, Dr. Rudolph Tanzi and Dr. Rob Moir. Dr. Tanzi is the Joseph P. and Rose F. Kennedy Professor of Neurology at Harvard University and Vice Chair of Neurology, Director of the Genetics and Aging Research Unit, and co-director of the Henry and Allison McCants Center for Brain Health at Massachusetts General Hospital. Among his many career highlights, Dr. Tanzi and his research team created Alzheimer's in a Dish, a 3D neural cultural system that has made drug screening for Alzheimer's disease faster and more effective. Dr. Moir is a neurobiologist and heads the research group within the Genetics and Aging Research Unit at Mass General Department of Neurology and is a faculty member of Harvard Medical School. Some of his many career highlights include uncovering new therapeutic targets and revealing the importance of metals in the pathological aggregation of beta amyloid. Now we're going to hear about their most recent work, which suggests that Alzheimer's disease could be an inappropriate response to a perceived infection in the brain. We're very fortunate to have both Dr. Tanzi and Dr. Moir join us today to discuss this exciting work, and with that, I'm going to turn it over to Dr. Tanzi.
Thank you, Missy. Can you hear me okay?
Yes, sir.
Great. Well, thanks for hosting this. It's great to be here. My colleague next to me, Rob Moir, will be doing the bulk of the heavy lifting here, but just in the way of introduction, you know, we've known about the amyloid beta protein now for 35 years, since George Glenner found it in 1984. And we've known about the APP gene since 1987, when we and others first published it. But since then, we know very little about what the APP actually does. We've always assumed that A beta is just junk, something to be eliminated from the brain. We've also learned from clinical trials that trying to hit amyloid beta protein in patients who already have symptoms, and even if it's early mild, based on the Biogen data, that if you try to hit when they already have symptoms, it doesn't work. Amyloid is a very early event. I know the press treats these trial failures as binary events. It's yes or no, or amyloid yes or no, or throwaway amyloid because the trial failed, but it's really not like that. These are not binary events, and we know that amyloid accumulates in the brain very early, 10, 15 years before symptoms, and if you want to hit it, that's when. The question is, and what's relevant for today, is when you do want to start hitting amyloid 10 years before symptoms, the way we hit cholesterol 10 years or 15 years before heart disease, or even earlier, should we wipe it out or just dial it down? The assumption has been that amyloid is just a byproduct of aging and accumulates in the brain like junk and garbage. But the fact is that we're learning from, especially from Rob Moir's work, we'll speak in a moment, that amyloid beta protein may not just be debris and junk, that it may actually play a role in the brain, just like its precursor, APP, has many roles in the brain. Sometimes we joke APP has been found to have so many roles that we call it APP as an all-purpose protein. And one of those roles is to make A beta, and for A beta to actually play a role in the brain as part of the brain's innate immune system. So what Rob's going to explain is a discovery that now is over 10 years old. I mean, I remember that day, that Friday afternoon, drinking Coronas at our beer hour.
Yes,
and at the same time, I was realizing, this was probably 2006 or 7, same time I was seeing association with CD33 and Alzheimer's, which is the first innate immune gene associated with Alzheimer's, unless you consider ApoE also an innate immune gene. But CD33 popped up, and I literally walked over to Rob's office next door and said, hey, so what do you know about innate immune genes? And he said, well, what do you know about innate immune antimicrobial peptides? And he was developing this Excel sheet that was about 5 pages long with uncanny resemblances between amyloid beta protein and known antimicrobial peptides. So for both of us, that was a revelation that the innate immune system matters. And the innate immune system here matters in 2 ways. One is producing amyloid beta protein in the brain, which, as you'll hear, may not just be a mistake, and also regulating neuroinflammation. So with that introduction, Rob will tell you the story now about amyloid beta protein as an antimicrobial peptide, and what this means also. We can talk at the end about what this means for therapies aimed at preventing Alzheimer's disease by hitting amyloid beta protein.
Yeah, thanks, Rudy. And Rudy and I have been bickering and working this out now for 25 years. So we've been developing these ideas for a long time. So what I'm going to try and do today here is, rather than just do a big diatribe of what the theory is, I'm going to sort of take you through the key findings.
Okay, can I interrupt you for 1 second? I'm sorry to do this. We've lost your presentation on the screen, so let's try to figure out real quickly how to fix that, because we had it before.
Why don't you escape here?
Okay, I'm escaping here. I'm back on the WebEx thing. Okay, I'll try and share Keynote. There we go.
Perfect.
Coming back, yeah.
Okie dokie.
Ready to go. Thank you.
All right, so.
We need to hit this.
Yeah.
presentation mode?
Yeah, it's in presentation.
There we go.
Okay, so what I'm going to try and do is take you through the key finding, the real nub of how we arrived at this new view of AD pathology. And that key finding focuses around the A-beta peptide. And so I'll take you through a brief history of Alzheimer's disease. In the past, it's always proved helpful if a quick overview of where the disease comes from and what the major pathology really is. And then I'll take you through A-beta and our discoveries that finally identified it as an antimicrobial peptide, an AMP of innate immunity, and then discuss a little bit about what the antimicrobial protection hypothesis, this new interpretation of the pathology in AD, what it means and how it can impact potential therapies. So we'll start. So, Alzheimer's disease. All right. First described 1906 by Alzheimer, and he had a patient, August Dutter, that he was looking after for a number of years. She had increasing dementia, and when she died, he looked at her brain at autopsy, and the first thing he saw was a clear degeneration of the brain itself. And you can see here on the right just how atrophied an Alzheimer's disease brain can get. It's clearly wasting away. So that's on the macro level. Clearly something's wrong. But it was when he looked at the microscopic level, down close at the brain itself, that he found the pathological features that would be the hallmark of the disease. And the first one was this: insoluble deposits, millions of them, of a substance he named amyloid. He thought it looked like something that comes out of plants. Turns out it isn't, but that's the origin of its name. And if you get even closer and have a very close look at these deposits, you see they have a very distinctive morphological structure, a core and are surrounded by more material. And that's a mature amyloid plaque. And they occur outside the neurons in the brain. And the second feature he noticed were these. These are long thread-like structures called neurofibrillary tangles. They're inside of neurons. Now, neurofibrillary tangles actually pop up in a number of neurological diseases, but amyloid plaques, they're specific for Alzheimer's disease. And so those two hallmark pathologies have defined Alzheimer's disease, and pretty much between 1906 and 1984, that's pretty much where our understanding of Alzheimer's disease was. But then, as Rudy mentioned, 1984, Glenner and Wong discovered what these amyloid plaques are actually made of. And they look like big solid lumps, but they're actually not. They're more like a big ball of tangled fishing line. They're made up of long fibrils. And each one of those fibrils is composed of a small protein called A-beta that stacks on top of one another to make these long, extremely strong structures that can become tangled and form this amyloid plaque. So we now know there are actually three pillars to AD pathology. The last couple of decades have seen the emergence of neuroinflammation as another key component. And so we have amyloid plaques, we have neurofibrillary tangles, and we have neuroinflammation. And increasingly, it's looking like neuroinflammation is the guy that does most of the damage. You can have a lot of amyloid and you can have a lot of neurofibrillary tangles, and you're still okay. But once neuroinflammation kicks in the later stages of AD, you start to see neuronal death. And the main cell that seems to be important for that is called glial cells, and they're the foot soldiers of innate immunity in terms of, if you imagine antimicrobial peptides are foot soldiers, immune cells like microglia are the heavy armor, the tanks. So it's called gliosis when these things start to mobilize in the brain. So the pathology sequence is this. First you get amyloid, that produces tangles, and together they produce neuroinflammation. So this has been characterized as the amyloid cascade hypothesis. And the amyloid cascade hypothesis has been the predominant model for Alzheimer's disease for nearly 30 years. And essentially it says it all starts with amyloid. And by the way, we still ascribe to that. It's just our findings have put a new interpretation of where amyloid comes from.
Or what triggers its deposit,
Yes.
deposition in the brain.
Why it forms. So this is a quick trip through a brain region, hippocampus, and it's actual slice from a brain, a 3D image. And as you can notice up the top there, these long strands hanging down, they're from neurons. And you can imagine they're like railway tracks. And the hippocampus is like a station that used to be under New York, where a switching station, where all of these different trains coming in from all the different parts of the brains come in and they get sent out to different regions. Well, in the brain, they connect to memories situated in different parts of the brain. And it's those that really get targeted Alzheimer's disease. And when you have memories, it's the impulses traveling up and back between those connections that really form and allow the thing to happen. So Rudy mentioned, or earlier on Missy mentioned, the Alzheimer's in a dish model. It was developed by in this unit by Du Choi. We published back in Nature in 2014. And this really has changed the way we do business. It's a fantastic model. It allows you to literally look in the brain. And essentially what it is a three-dimensional growing of human neurons. And Du has improved it continually over the last few years, and it now contains many of the cells that you find in brain. And it allows you to see what's happening literally down at microscope, something you just can't do in a living brain. But it also allows you to screen for drugs, and Rudy will talk about that later probably, in what is would normally take a couple of years and thousands of mice. You can do in a month or so at probably a tenth the cost. So it's really big. But what we've been using it for is to study amyloid generation. And here is just a sort of example, a movie showing you these neurons growing, connecting, and there you have this entire network. And I'm sure it's going to start thinking soon, these things. And it's full of most of the cells you find in normal brain, including those microglia. And what happens when you get amyloid deposits is that microglia are normally caregivers. There's about 10 times as many of them as there are neurons in brain, and they tend the neurons. They look after them. They help them. They are there for support. But they're not just caregivers. They're actually immune cells, and you can flip them into warrior mode. And when they do that, they become soldiers, and they will attack all sorts of things. And when you get the presence of amyloid, lots of it, as you can do in this model, you see this. And I just watch where these arrows are and watch the red cells.
Let me just mention that this this video comes from the new Alzheimer's in a dish model that incorporates microglia with microfluidics that was developed by Han-Sung Cho at UNC Charlottesville, and also Joseph Park, who just joined our unit about a year ago. And so this is the next level. The first Alzheimer's in a dish had plaques and tangles. This one includes now visualizing neuroinflammation in the form of recruitment of microglial cells and then their conversion into these warrior killers that you're about to see are going to digest neurons and synapses.
That's right. And as I said, they're caretakers or caregivers as long as things are going nicely. But when you start to get a lot of amyloid, this happens. And if you'll see on the right-hand side, see that red cell? Attack. This is worth watching again. It's just an amazing picture. This time, if you watch on the left, you'll see how that long green neuronal projection is snapped and springs back.
Right, which meant probably either a synapse was pruned and it snapped back, or could have been an axon cut out of the focal frame of the video, so it causes that axon to snap back.
And if you remember back to the previous movie where I showed you those long dangling projections that connect different parts of the brain and allow memories to be retrieved, well, that's what these things are. And watch what happens to them here. Snap. So effectively, that's what's happening in Alzheimer's disease. Those long projections that connect neurons and connect different parts of the brains are getting broken. And so in a very real sense, your memories are still there. It's just you can't access them. The connections are being destroyed by this neuroinflammatory response driven by amyloid deposition, which drives these cells crazy and turns them into the warriors. So that's the sort of pathological model that's evolved in Alzheimer's disease now. And genetics have really led the way in understanding AD. It's really genetics that has shown us the way towards understanding this process. And it also shows that A beta, despite a lot of what you hear at the moment, is the key to Alzheimer's disease. So mutations in the A beta.
Let's just say the key trigger.
Yes.
But it's the neuroinflammation that we just visualized.
That's
doing the bulk of the cell death. But arguably amyloid, which is causing tangles and cell death, is what's causing the conversion of microglial cells to convert from, housekeeping cells to then cells that are trying to wipe out a part of the neural network that these glial cells, by seeing neuronal cell death, are assuming there's infection.
Yes. So mutations in A beta produce early onset Alzheimer's disease. So people get it before they're 65. And this is concrete evidence that A beta is right at the heart of this whole process, because it's A beta generation that's being modified by these mutations. So, of course, familiar Alzheimer's disease, it's less than 2% of cases. So the other area that genetics has been absolutely key is to produce or reveal risk factors for non-familial AD, the most common form, the form that 98% of people get. And the risk factors fall into a number of groups, and prominent among those groups are the innate immune proteins. So many of the genes are genes that are involved in innate immunity. There's also, of course, genes involved in the direct processing of A beta, but really there's a lot more. This is actually a fairly old slide now. There's a lot more innate immune genes linked to AD than is shown here. So innate immunity is the ancient arm of the immune system. It's not the one you usually hear about, antibodies and vaccines and things like that. That's the adaptive immune system, and that works in the periphery, outside the brain. But adaptive immunity it's a fairly rough player. You can't really have that operating in the brain. It's a very delicate structure, and so it's left up to innate immunity to do most of the protection there. Now, innate immunity is very old. It's what protects cockroaches and worms and flies, and it's still key in our immunity as well. It really is the foundation upon which our more sophisticated adaptive immunity is built. And in brain, it's the only game in town. And the main mediators or the main foot soldiers of innate immunity are antimicrobial peptides. They do most of the heavy lifting. And then you have the more, as I said, analogous perhaps to the heavy armor, the tanks. They're the cells, microglia, and astrocytes. So, the progression of AD now appears to be this. In the first line here, the brown line is in fact amyloid deposition. And you can see amyloid deposition, it starts 15, maybe even 20 years before you have any signs of cognitive impairment. And by the time that you're actually showing mild cognitive impairment, amyloid deposition has pretty much peaked, actually. But that has triggered the next major pathology, which is neurofibrillary tangles. And as mild cognitive impairment, MCI, starts to take off, you start to see a lot of neurofibrillary tangles. And as the dementia really starts to set in, that's when you see a lot of neuroinflammation. And so those two previous pathologies, there's what's driving the neuroinflammation. And this is a very good graph because it also shows you really why most of the anti-A-beta therapies probably didn't work, because you really have to hit A-beta well before you're showing any cognitive impairment. As Rudy likes to say, it's like you strike a match and set fire to a forest, and then you blow the match out and expect the forest fire to disappear. It's just not going to work that way. If anti-A-beta therapies are going to be effective, you're going to have to be able to treat people well before they're starting to show dementia and identify them.
Or target what is striking the match.
Yes.
And I think that's going to get to the heart of the talk here.
Yeah, and I'll talk about that a bit further on.
Yeah.
But that suffers the same problem. We don't know who those people are that are starting to develop heavy amyloid loads yet. And we're working on ways to try and identify them, but at the moment, I think I would say that Rudy would agree, most of the anti-A-beta therapies are on the shelf until we develop techniques
I wouldn't. I mean, I think the press wants you to believe that we're throwing the baby out with the bathwater here.
I didn't say they were gone.
No,
I just said they're on the shelf.
But remember that in February of 2018, the FDA, namely Scott Gottlieb and Billy Dunn, released new guidelines for Alzheimer's that said if you have a drug that safely moves a biomarker we all believe in, like amyloid, and it's safe, they will consider approving it and then look for what's called real-world evidence or real-world data once the drug's approved to see if it's actually preventing Alzheimer's 5 or 10 years down the line. This is a huge step for the FDA if they go through with it, because frankly, no pharma company is going to do a 10-year prevention trial that costs $5 billion, and then when you're done, and if it works, your drug is so far along that your drug's now off patent and a generic. So, you need this type of progressive thinking that the FDA is showing. And so I think a lot of these amyloid treatments that have failed, for the ones that are safe, economically feasible, that can be used, the way we use statins to hit cholesterol, we can bring them back. Of course, for immunotherapy, they're expensive and there are side effects. So we may have to wait for a little white pill that does the same thing. But you can imagine that a safe antibody that hits amyloid, if somebody has a lot of it in their brain on an imaging scan or someday a blood test, that you might want to pull that amyloid out, more rapidly, and then that might warrant using an antibody approach. So this is all yet to be seen in the future, but certainly we're not going to throw away amyloid because it failed in symptomatic people again, like it did every other time.
Yeah, no, I agree. I said it was on the shelf until we make some more advances. They're not driving hard at amyloid.
To a different shelf.
Yeah.
Yes.
Yes. Yeah. They're not driving hard at amyloid anymore. And particularly neuroinflammation is starting to become a big target. So, amyloid, tangles, neuroinflammation. So, the dominant hypothesis for AD for 30 years has really been all about A-beta. And essentially it says this: A-beta is a functionless piece of metabolic junk that spontaneously forms amyloid without a physiological reason. It's just a bad boy. In essence, A-beta is intrinsically bad. Nothing good about it. And you don't need anything else besides this A-beta peptide, which will just, for its own weird reasons, suddenly form fibrils and cause the whole process of Alzheimer's disease. The trouble is, that kind of was a reasonable idea when A-beta was first discovered, 35 years ago, because everybody thought it was only found in AD, and also the way it's made was thought to be a very abnormal process. But it turns out that isn't an abnormal process, and it's not just found in AD. So there's a lot of data that no longer supports this idea that A-beta is intrinsically a bad boy. And a conspicuous example of this, I think, is this. For example, A-beta is generated in nearly all vertebrates: birds, reptiles, frogs. And the human A-beta has remained completely unchanged for 400 million years. That human A-beta is found in over half of all vertebrate species. Now, if it was bad and useless, it would have disappeared a very long time ago. What this screams to us, anyway, is that A-beta is doing something important, and it's highly optimized to do it. And that ability to form fibrils is almost certainly at the core of it. Now, it's not been clear what that was until, as Rudy mentioned earlier, we serendipitously stumbled across antimicrobial peptides. And the similarities between A-beta and antimicrobial peptides, AMPs, was just jaw-dropping. And as he said, I really did have a spreadsheet with 5 or 6 pages just listing the similarities. So that was over 10 years ago, and the first thing we did...
You may have spent a little too much time on that Excel sheet.
I might have spent it, yeah, I know, but I wanted to be right. Because I knew it's a radical idea in the field. And so you want to get your ducks in a row. So the first experiment we did was a very simple one. We just mixed amyloid A-beta and bacteria to see whether it would inhibit their growth. And this is the figure showing the assay. It's called a button assay because where there's a growth of bacteria, you end up with a little button at the bottom of the well. Where there's no button, no growth, A-beta inhibited it. So as you add increasing amounts of A-beta, you see the buttons disappear. And this is a very powerful, simple tool. It's like something that comes out of the 1950s, but it's still used today. And it showed very nicely that A-beta is actually a very potent antimicrobial peptide. In fact, it's about 100-fold more potent than penicillin for some pathogens. And that really got us convinced that we were on the right track here, because that kind of activity is extremely rare. People have generated millions of synthetic peptides with the idea of making an antibiotic that is does the job of antimicrobial peptides, except to use it clinically. And nobody has yet made one with the kind of potency that we're seeing here. So the next question, of course, is, well, that's fine in a test tube, but does it do that in live animals? And so for the next several years, we developed 5 models to show that expression of A-beta, human A-beta, will protect you from infection. And so you've got cultured cells, we've got little nematode worms, we've got Drosophila, which is fruit flies, and of course mice. Now, this is the only graphs I'm going to show, and you don't need to really see. All you need to see is that in all the cases, A-beta protects. But the really interesting thing is how it does it, because A-beta is an entrapment amp. And you can see here in this really nice photo that amyloid fibrils ensnare the bugs, trap them and wrap them up and essentially neutralize them by entrapping them. And when I said the A-beta plaques are like fishing line, tangled fishing line, that's actually closer than you'd think, because that's the sort of way that it works. It works like fishing line. And it pulls them in to clumps. And in those clumps, they're pretty much neutralized. It also means in those clumps, well, this is some examples. It does it with salmonella, that's bacteria. It does it with viruses. It does it with fungal infections. Same mechanism, very effective. In those, here's a few other nice pictures showing those fibrils entrapping yeast, bacteria, and viruses. And it's all the same mechanism. These very, very strong fibrils, we're talking, Kevlar type strength, can wrap around the bug and pull them in to these aggregates. And inside those aggregates, those amyloid plaques, A-beta's killing activities start to come to the fore. And one of the things they do is they poke holes in the membranes, essentially causing the microbe to bleed to death. They do another really neat trick, generate a huge burst of bleach. And the bleach sterilizes the plaque. It also kills the peptide's ability to make more bleach, which is pretty important because you wouldn't want that going on for years. It chemically alters the peptide itself. So it's a self-limiting type of system. It also causes the chemical cross-linking of the fibrils, which literally welds the cage shut. So if there are any surviving microbes, they're not going to be able to work their way out. So this is a good picture. It shows a close-up of bacteria trapped in an amyloid plaque, and there's the amyloid fibrils. And if you can see that the cell looks all shrunken, and that is because of these effects, these killing activities. And if you look in a living or a mouse brain, you see the same thing. Amyloid trapping microbes. The microbes, the salmonella here, are green, the A-beta's red. And I like this picture particularly because it's right at the cusp. You can see that the bacteria are just starting to overwhelm the amyloid. The amyloid pool is exhausted, and the bacteria are starting to get the upper hand. This mouse is actually clinically in pretty good shape, considering the incredible load of bacteria it's got in its brain. If it was a normal mouse that didn't make human A-beta, it'd be long dead. But in an hour, maybe two, this mouse is going to go into rapid clinical decline and die because the amyloid protection has been exhausted. So this is an acute infection model. That means that they get very sick and die. But really for Alzheimer's disease, we're looking more at chronic infections, long-lasting ones. And if you infect a mouse with herpes simplex 1, which is a very common pathogen that's been linked to Alzheimer's disease, it gets sick for a week or so, then it recovers, but the virus is still there. The virus will be there for the rest of the life of the mouse, slowly ticking away. This here does not look like an amyloid plaque, but if you look in the brains of those mouse with chronic infections, a few weeks later, you see that. And that looks like that. The thing that Alzheimer saw when he looked down his microscope over 100 years ago. And at that point, I would say this whole antimicrobial peptide identity for A-beta has led us to believe that Alzheimer's disease is the result of an innate immune response to a perceived infection. Now, the infection may be real or it may be falsely identified, but I mean, there's a lot of data now mounting that suggests that microbes are indeed involved in Alzheimer's disease. It's not as simple as one microbe gives you the disease. Koch's postulate which has served us well for 150 years, says one microbe gives you specific disease. But for Alzheimer's disease, it's looking increasingly like it's not quite like that. And that essentially is the antimicrobial protection hypothesis. And at this point, I'm going to hand it over to Rudy to say a few more things.
Yeah, I mean, I think you make an important point. You know, when we talk about infection, but this is subclinical. This is subacute. It's not an infection that would be clinically noticed. You know, I think the result that really blew my mind was in the Science Translational Medicine paper in 2016, where Se-Hoon Choi injected salmonella bacteria into the brain into the hippocampus of a very young Alzheimer's mouse, only 4 weeks old. So it had no plaques, and it wouldn't have plaques until about 5 months old. It was a 5XFAD mouse. And we saw full-blown plaques littering the hippocampus overnight. And since the salmonella were fluorescently labeled, you could see one bacterium was able to generate an entire plaque overnight. And so the paradigm's always been it takes 10 years for a plaque to form, only taking into account production of A-beta and clearance of A-beta. So if you remember the work that Conrad Bywater and Colin Masters used to show, here's how much A-beta is made per minute, here's how much is cleared from the brain, here's how long it takes to form a plaque, based similarly on completely on plumbing, 10 years. But it didn't take into account that amyloids are nucleated or seeded and can be nucleated rapidly. And those microbes were rapidly nucleating amyloid overnight. So I think one of the things that came out of this hypothesis is change in the paradigm. And this agrees with Brad Hyman's old popcorn plaque hypothesis from years ago. The plaques can form rapidly and be nucleated rapidly. So you have to ask what nucleates them. And if you look at early onset familial Alzheimer's, A-beta 42 does. You don't need a microbe. If you have an early onset familial mutation, an APP or presenilin 1 or presenilin 2, that increased ratio of A-beta 42 to 40 is enough to just nucleate plaques all the time from birth. But 98% or 99% of patients do not have early onset familial mutations. So we should be asking what nucleates, what seeds plaques in that case. And here's where microbes make great candidates, that microbes are seeding the plaques. Then, the next step is neurons start to die and glial cells react with neuroinflammation. So in some ways you can say that's silent infection, that the innate immune system's actually reacting to the plaques and the tangles and the neurons that are dying. But on the other hand, if you say that microbes triggered those plaques or nucleated those plaques in the first place, then it's a new version. It's not quite sterile infection. It's not quite infection. It's microbes eventually driving enough neuroinflammation to kill enough neurons to get demented, but doing so by the microbes precipitating or seeding amyloid first and then getting that cascade of plaque and tangle formation. And finally, I just want to say that, the other reigning paradigm in the amyloid cascade hypothesis is that it takes a very long time to go from amyloid to a tangle. And I think to some extent that was a hand-waving argument for the fact that Alzheimer's mice don't get tangles. You put plaques in and they don't get tangles. So the assumption was made, hey, they just don't live long enough to get tangles, and it takes a long time for amyloid to cause a tangle. And you have to go through all these steps, including oxidative stress. Well, in the Alzheimer's in a dish model using human neurons that express the right forms of tau protein, tangles form immediately from amyloid. You have amyloid made in the dish, and then a week later you see plaques. So it's not a long process. So I think we have to also consider that two reigning paradigms, that it takes a long time to go from a plaque to a tangle, is probably not right. And the other reigning paradigm that it takes 10 years to form a plaque is probably not right. And that microbes may play a key role, or maybe other nucleators, maybe air pollution. But anything that can seed amyloid rapidly may be triggering this disease or plaques and tangles. But ultimately, it's what we call the silent infection of neuroinflammation responding to dead neurons that's going to cause enough cell death to get to dementia. So this gives us a lot to think about in terms of drug strategies. I think in patients with symptoms, you need to hit neuroinflammation. It's too late to hit plaques. But if you want to nip this disease in the bud, hit those plaques early on, know who's at risk, and hit them 10 years before symptoms, as shown in this slide. But if you want to go even earlier than plaques, you might want to hit the microbes that are seeding those plaques in the first place, or maybe air pollution or other particles. But we need to think more about seeding of amyloid. And I think even with ApoE, we've also assumed E4 versus E3 works by clearance of amyloid from the brain. Again, the plumbing hypothesis. How much do you produce? How much do you clear? But there's increasing evidence that ApoE4 can precipitate the seeding of amyloid. In fact, Rob, we had a paper in 2005 that showed E4 actually seeds amyloid production better than E3. So seeding is becoming a key here, and the role of microbes in seeding amyloid rapidly, I think, has to be a major therapeutic target going forward.
Yeah, and I would say that, in essence, the antimicrobial protection hypothesis says that Alzheimer's disease arises because of at the interface of the innate immune system and a perceived challenge, whatever that challenge may be. It's that point where those two things meet, which starts the disease process.
But it means also that amyloid is working together with glial cells as part of the innate immune system.
Yeah.
And they're also connected. A microglial cell tries to clear an A-beta oligomer, and if it can't, it gets activated to become pro-inflammatory and producing cytokines. So there's a lot of interaction and crosstalk between the production of amyloid, the deposition of amyloid in terms of being seeded, for example, by a microbe, and then finally the interaction with microglial cells being activated to cause the degree of neuroinflammation necessary to kill enough neurons to cause dementia. So I think we should probably stop our comments and maybe we take some questions now.
I think so.
Is that right?
Yep.
So Q&A.
Great. All right, great. So I'd like to just remind folks to use the chat feature at the bottom of the screen to submit your questions. We do have our first question, and that was, how far off is a blood test?
Yeah, so at least two groups are doing a blood test. Right now you can pay several $1,000s out of pocket to be imaged. Healthcare won't cover that. It's FDA approved. Healthcare won't cover it because they say amyloid's not yet actionable. Well, in fact, it is actionable, but those therapies aren't approved yet because they're being forced to have effects on cognition. That might change with the new February guidelines from last year. But the blood test, Randy Bateman is developing a blood test that's moving rapidly at WashU. Cure Alzheimer's Fund is actually funding Randy Bateman and doing great work. He just won the Potemkin Prize, which is the biggest prize in Alzheimer's now. And also there's a collaboration between Colin Masters in Australia and the ABLE project, that's like ADNI in US, together with folks in Japan who are developing another blood test that's using mass spec. But I think we're going to have a pretty cheap one to be used by diagnostic companies like Quest and the like. And I think Randy Bateman is probably closest to that. And I don't know the timing on it, but it could be within, if things go well, it could be within a year or so.
Okay, we've gotten a few more questions here from Bruce Pyenson. A European study used real-world data to suggest that patients treated with isoclover had lower incidence of AD given the long duration and multiple potential causes. How can real-world data help with this important work?
Yeah, it's a great question. I mean, I think we can think about real-world evidence and real-world data as being the future of not just coming up with new clues into the etiology and pathogenesis and factors involved with disease, but also for clinical trials. If the FDA actually does allow safe drugs that move amyloid, if they allow the approval of those and then track real-world evidence or real-world data that they're working, I think it's going to be best for all of us. I think we can also make use of epidemiology data, biobank data, UK Biobank. We have a biobank here at Mass General, patient registry data. We need to do much more deep machine learning type of searches of patient records, electronic health records, to see what we're missing, especially in the way of infections and antimicrobial medications that might be preventing Alzheimer's. Maybe, Rob, you can explain what happened with the Taiwanese study on herpes.
Yes, so we're actually initiating some of that ourselves here with using Mass General and Partners databases. But there was a big study that came out last year out of Taiwan, 30,000 people, a lot of people, and they were looking at herpes simplex 1 infections. And what they found was that people with chronic herpes 1 infections had a 2.5 times increased risk for Alzheimer's disease. And mild infections, they still had an increased risk. It was about double, or no, it was 1.5 or something like that. But the real kicker was that there was several hundred that had been receiving anti-infective treatments, anti-herpes medications, and those people, their risk of Alzheimer's disease returned to baseline. Now, returned to baseline, didn't go below baseline, just went back to what everybody else's risk is. So that study fits very nicely in with our sort of findings that these microbes can accelerate amyloidosis and thus push the disease a lot faster. And so that's an excellent example of how these big data studies can really inform us. And there's some key features about that. But I would also emphasize it returned them to baseline. It didn't eliminate the disease.
Yeah, I think it's important because, that doesn't say you can just take an antiviral and try treating Alzheimer's or patients. This is a case where you had a herpes trigger of amyloid that you're hitting early on with an antiviral and help nip it in the bud. So you got to always put these different types of drugs, right patient, right person, right time. Yeah. Okay.
Okay, we have another question from Stanislava. Infection likely induces expression of other AMPs. Do you think that other AMPs interact with amyloid beta in the course of AD to kill neurons?
Yes, and we're actually working on this pretty much now. There's another important antimicrobial peptide called amylin, which actually is involved in type 2 diabetes. It's not generally recognized, but diabetes is an amyloid disease, and this peptide amylin does the same thing in the pancreas that A beta seems to be doing in the brain. And it's a very, very potent antimicrobial peptide. And we find that in amyloid plaques, amylin is turning up there as well. And their effectiveness is synergistic. So if you put both of them together, they work much better than either would on their own. So that's another complication to the whole process. And I think there may well be other antimicrobial peptides that are hooked up with all of that too. So how important amylin will turn out to be in Alzheimer's disease, I don't know. I think it's going to turn out to be critically important in diabetes, and for the same reason that A beta is in Alzheimer's disease.
It's important to note that amylin is one of at least a handful of antimicrobial peptides that can form amyloid if unregulated. So you have amyloid in the eye and the heart and the seminal vesicles, all of which involve what are otherwise antimicrobial peptides that have gone a little too far in their extracellular trapping of microbes and now are causing amyloidosis that challenges organ health. And essentially, you can say the same thing about plaques and the brain using that model.
Yeah, there are probably, there's over 30 amyloid diseases known, and about 10% of them are caused by known classical antimicrobial peptides. So there's plenty of precedent for A beta being an antimicrobial peptide that's doing more than it should, perhaps.
Okay, we have another question. For people who develop AD, are their amyloid levels higher than normal? Would regulating amyloid help prevent overaccumulation of A beta and plaques? And can amyloid level be tested for?
Well, yeah, this gets back to the blood test earlier question. So we can visualize amyloid in the brain by imaging, and I think soon there will be a blood test, maybe more than one, to get an idea of who has high amyloid. So you might imagine in the future that at some age, let's say by 50, not only do you get your colonoscopy or your breast cancer exam, but you also check for amyloid in the brain, perhaps with a blood test. And if your percentile is too high, then you would start an anti-amyloid therapy. If it's very high, you might have to use an antibody to bring it down and then maintain it with something like a gamma secretase modulator, or maybe if there's a BACE inhibitor that turns out to be safe. But that would be a scenario in the future.
Okay, we're trying to determine if some of the questions that are being posted are already answered. But I'm going to throw out this one, and if you've already answered it, and this is the second question from Bruce Pynton. Are there analogs to A-beta antimicrobial peptides in the innate immune system of bacteria?
There are regions that certainly look similar. Herpes has one. And antimicrobial peptides, bacteria use them against one another all the time. Is there one that looks like A-beta? Sort of. It's a fair way away, though. And there are certainly entrapment amps that bacteria use against one another. But most of them are not, they're not A-beta. They have many of the physiochemical characteristics, but they're really not the same peptide. A-beta's got a few interesting little quirks to it, such as its ability to bind metal ions, and that's a kind of vertebrate thing. Most bacterial amps don't bind metal. They don't need to. It's dangerous for them to do it. But we get benefits from it because we can generate bleach with amps that bind metals can generate bleach, which is the most toxic antimicrobial molecule known. So it's a little bit different in some ways from bacterial amps.
All right, and I have one last question from John. I am diagnosed with what is thought to be atypical Alzheimer's frontal variant, in parentheses, because of a research autopsy on my father, whose path was very similar, and he had AD. Are there theories on why some types don't start in the short-term memory?
Look, there are different types of dementia. First of all, I'm sorry about your troubles, John. There are different types of dementia. Alzheimer's generally begins, with more of the hippocampus, entorhinal cortex, short-term memory, but you can have frontal lobe dementias that begin in frontal lobe regions, or you can have Alzheimer-like dementias that begin in other parts of the brain. And so, it sounds like maybe this was a case where dementia or damage began in the frontal lobe or prefrontal cortex. And in any case, these pathologies spread. So if you have Alzheimer's with short-term memory changes, your long-term memories may be preserved at first, but then eventually the disease will spread. So there are different variants of dementia along the Alzheimer's spectrum. Although the most common form of Alzheimer's you see mainly affecting first the hippocampus and entorhinal cortex, just that region.
Yeah. Alzheimer's is about 4 out of 5 dementias, but that still leaves a lot of other diseases.
Do you have time for one final question?
Sure.
Okay, from my colleague Ryan. He wants to know your thoughts on combination therapies moving forward. Well,
yeah, I think most would agree that combinations will be best. You might want to target, in order of chronology, you might want to target a microbe or some other event that's triggering amyloid deposition. For example, my colleague Steve Wagner and I developed gamma secretase modulators, not gamma secretase inhibitors, which are not safe, but gamma secretase modulators that reverse the ratio of A beta 42 to 40, because the mutations increase that ratio. And that's something you might do early to stop seeding of amyloid by A beta 42. You might target microbes that seed amyloid. You might target the clearance and aggregation of amyloid or tangles. If you just go through the pathway, you can come up with different ways to hit the disease. But when you're thinking about combinations, you still have to keep in mind using the right drug in the right person
at the right time.
At the right time. And so, if someone already has symptoms, it's probably not going to do much to stop the aggregation of amyloid, as we've learned. But it wouldn't hurt if you were treating, for example, neuroinflammation process in a patient, and that's the main thing they need. It wouldn't hurt to add an amyloid aggregation drug as well. Just like in heart disease, if someone has congestive heart failure and a heart attack, the cardiologist just doesn't say, here, just take a Lipitor and go home. You have to do something about fixing the heart. But they might take Lipitor anyway, or some type of statin to keep their cholesterol down. But in terms of what's going to actually help at the right stage, it could be very different drugs. But they, for example, with amyloid, if you're treating amyloid early on, a combination of drugs like BACE inhibitors, gamma secretase modulators, and drugs that promote microglial clearance of amyloid, like, the drug Chromalin that AZ Therapies is developing. You might look at that combination. But, combinations are probably going to be the way we go, but we still have to keep into account personalizing according to best drug for the best person at the right time.
Okay, well, thank you so very much. We have thoroughly enjoyed having you on today, and we've just gotten so many questions. We unfortunately can't possibly get to them all, but we do plan to make the webinar available shortly on the act-ad.org website. And we want to thank all of our participants today for your time and thoughtful questions. So thank you so much to Dr. Tanzi and Dr. Moir for participating.
A pleasure.
Yeah, very welcome. Thank you.
Thank you. Bye-bye.